Our last talk is by Dr. Todd Holland, who is a neurosurgeon at the University of Michigan. He's an assistant professor of neurosurgery. His work hopes to be part of the next generation in which computation and machine learning will be made a part to be included in scientific breakthroughs. His talk today is entitled Rapid Intraoperative Molecular Classification of Gliomas Through Optics and Artificial Intelligence.
Great, thanks for the opportunity to speak, and excellent speakers that preceded me. It's good to see that we're hearing a lot more about machine learning, AI, and deep learning, and this is going to be kind of an extension of what you've already heard about how we can really use imaging data sets to do molecular classification. So I just have one disclosure. I am a shareholder for the company that makes the microscopes that I'm going to be talking about. So conventional brain tumor diagnosis really requires multiple modalities, and we're still reliant on that, where we're doing some histologic analysis combined with some set of molecular diagnostic tests, and that can be, IHC, FISH, sequencing, epigenetic assays, methylation studies. And really, there's a multifaceted set of barriers that may limit a patient getting all of these tests when they potentially need them. And one of the things that we're trying to develop in my lab, in collaboration with Dr. Oringer as well, is how do we streamline this pathway so it's easier for patients to get these diagnoses using a single system. And really what we're proposing is that any diagnostic system requires at least two things. When you strip all of them down, there's really two things. One is we need some data, right? Whether that's sequencing data, imaging, whether it's analog, microscopy images, whatever it may be, MRI. And the other part is analysis or an interpretation of that data. And what we're proposing is that the way to build the best, in my opinion, the sort of most likely way to get there is through optical imaging and artificial intelligence. And this is the question: can we develop a streamlined pathology workflow using a single diagnostic modality that is timely, efficient, and accurate? So this really starts with the imaging portion. This is the measurement portion, and the translation of the optical imaging technique that we use, called stimulated Raman scattering microscopy, has really been a true bench-to-bedside journey. It's well over a decade, and it's resulted in multiple publications, and it's really been a fantastic privilege to be part of this clinical translation that's been a lot of groups, including Dr. Oringer. And my contribution has really been how do we develop specific AI algorithms to help interpret these images. So this is the general workflow, and this is what we have already published. And a patient comes in, has a new brain tumor. We biopsy that brain tumor in the operating room. We're able to load that specimen into a stimulated Raman histology imager. We acquire an image, an optical image, label-free, so there's no staining of any kind, there's no processing, it just is a piece of tumor into the slide and into the microscope. We're able to generate large images, usually 3 by 3 or 5 by 5 millimeters, and then what I have been involved with is developing specifically deep neural networks to help with the interpretation of these images. And you've already heard a bit about patch-based classification, which is what I'm going to be referring to, and this is where individual regions within the image are used to train deep neural networks to perform some classification task. And we have developed specific ways to do inference where we don't care so much about patch level prediction, we care about patient level prediction. And we were able to develop a simple way to pool the probabilities from each individual patch in order to be able to make a final histologic diagnosis. And these are the results. We did a prospective trial. We're really big on the idea of doing prospective and external testing, and we were able to do that in a 2-armed non-inferiority trial where we used a conventional H&E diagnosis with humans interpreting frozen sections versus, and the other experimental arm, which was SRH, and then the model, the AI model that we had trained. And we trained this for the most common brain tumor types, and we were able to achieve our primary endpoint, and overall accuracy was comparable between humans, and also the mean class accuracy was very close as well, which is an important metric because that's more likely to show if you get errors wrong in rare diagnoses. So this talk is really an extension of what we've done in the past, and this is about how can we extend this general diagnostic framework to include molecular classification of diffuse gliomas, right? And this is a much harder task because, as you've heard, now we're talking about mapping pixels, in this case digital images, to molecular subtypes. And there's no specific reason why we should necessarily believe that there's going to be visual feature differences that result from individual tumor mutations. And that's the hard part. That's like the superhuman task that everyone has been talking about and we're all trying to model to figure out. And this is something where I think AI really shines, is where we can actually develop computer vision systems that can do things that human visual systems potentially can't do. So here's, again, another kind of framework, general workflow figure where we have our imager. This is the newest iteration of our imager that we're using in the operating room. We're able to generate our images as we normally do. And then there's two phases to this. There's a vision component and there's a classification component. The vision component is really about how do we go from images to image features. And we have a specific way that we train our networks that's different from our previous work. So you have to get an image down to a fixed feature vector, and then from there you have to do classification. And the majority of this talk is going to be about how do we leverage data we already have, specifically TCGA or sequencing data, to help do better vision classification of these images. So this is the way we train our deep neural networks for visual feature extraction, and we used something called contrastive learning, and specifically this is patch-based contrastive learning, which is, in my opinion, a more high-powered way to train these neural networks compared to a lot of the conventional ways that you read about in a lot of the image classification literature. We've shown that this works very well for patch-based classification. It probably works well for MRIs, and we've been looking into other applications of this specific framework. And when you do this specific kind of visual feature learning, you can look at the representation of these images in high-dimensional space, and then you do what's called t-distributed stochastic neighbor embedding, where you project these images, now these image vectors, into low-dimensional space, and basically clustering is good. If everything is all spread out and mixed, that's bad. And if you use our techniques, visual feature learning techniques, we improve upon the standard way to do everything, which is cross-entropy. But the point that I want to focus on is that how do we use prior knowledge about known molecular subgroups to help classify diffuse gliomas, right? We know that these mutations are not individual events, that they do tend to co-occur. And you can train it without doing that. You can just say, we're just going to train it, we're going to treat every event as independent, and we're going to train the models that way. But that to me didn't seem like the right thing to do, being that we know that these aren't independent events, and we know that there are well-defined molecular subgroups. And the techniques that we use really come from natural language processing, which we haven't heard a lot about, but specifically word embeddings. And this is about how do you represent words in computers, right, using binary. And that's kind of a strange thing to think about, but the point is at some point, if you want to do some kind of semantic processing, you have to represent a word using really either binary or in a continuous space, so in a discrete or a continuous space. And one thing that's been known extremely well in the natural language processing literature is that if you can learn to embed words in a meaningful vector space, that will significantly improve your ability to interpret sentences and produce meaningful answers when asked questions to natural language processing systems. And these are vector spaces here where not only are the points important, where you're converting words into vectors, but also the direction that you're moving in that vector space is also meaningful, that represents concepts. Things like royalty or femininity or any of the things that we normally think about when we're actually producing sentences, you can actually learn these vector spaces that will encode that. And one way to code it is using glove embeddings, and that's what we used. But the idea is download the internet, literally every word that's been printed. You look at all of it all at once, and you say, how often do words co-occur within some window, right? You say, how often within 50 words, how often do you see these two words occur? You populate a matrix. That matrix is the length of every word in the English language on the X and the Y direction. And each sort of cell in that matrix is going to be populated by a co-occurrence value. And if you do that, there's a really efficient way to learn the word embeddings such that that vector space is now meaningful. Okay? This is all published. This has been done. And we know that this works extremely well. And what we did in our group was use the similar idea for patients and their specific mutational status for their tumors. So use TCGA and CCGA and any other data set you can find, pull that together and say how often do specific mutations co-occur, and let's use that information to learn a vector space that not only the individual mutations are meaningful, but the direction in that vector space is meaningful, okay? This is the equation down here. That's a formal expression of it. So this is actually what you get. On the left-hand side, the word is the actual location in this learned vector space. And if you look in the circles, you can kind of see. So red, you see these different genes clustering together. The black is basically all the wild-type mutations. And in green, blue, and red, basically traversing in that direction in that space is going to correspond to known molecular subgroups, so oligodendroglioma subgroups. So that's IDH mutant 1p19q versus astrocytoma, ATRX mutation that co-occurs with p53, and then all your sort of different flavors of GBM mutations that come together in differing frequencies. But the point is that this works. We're able to do this reproducibly. And now, how do we use this to help classify images, right? Sorry, this is just the description of those subspaces. And again, not only are the locations meaningful, but also the directions are meaningful. Okay, so here's the sort of molecular classification framework that we're proposing. This is the kind of end-to-end way to think about it. We take an image, we train a neural network using the patch-based contrastive learning that I mentioned before. That's going to give us a feature vector. When we're training these, we can actually use the label embeddings, which is the gene embeddings that I had just mentioned. And then there's also an additional thing of state embeddings, which is to say whether the gene is mutated or not. We then aggregate that information using a transformer model. Don't worry about what that is, but it's another model that you can use in deep learning. And then you can output the prediction, individual predictions for each one of the mutations that you're interested in using both the molecular data, the vector space that I mentioned, as well as the visual features. So this is some of the preliminary results that we had. This is a mix of patients both from NYU and Michigan, and we were able to get excellent prediction on both canonical and non-canonical IDH mutations. So you can see the predictions in the second row in are the actual model predictions, and then we have the immunohistochemistry combined with NGS, and then the final diagnosis. And this is kind of what you get in the OR. So this is the segmentation in red is sort of the hand-labeled segmentation, and then we're actually able to automatically identify regions of dense tumor infiltration. Once we do that, those are the images that we predict on. This is really a validation set. These are University of Michigan patients, 50 patients that we are sort of a rolling group of validation patients that we're predicting on as validation, and we were able to achieve excellent precision recall curves using different models where we're using the simplest to increasing complexity up to the transformer and the gene embedding model. This is a multi-label classification task, so using one statistic doesn't work very well. You have to somehow collect these statistics together so you can compute mean accuracy, mean AUC, subset accuracy, et cetera. But we're still seeing consistently that our model is performing very well on these multi-label classification metrics, in this case for IDH, 1p19q codeletion, and then ATRX. And then I actually did this analysis this morning. So out of principle, we basically say if we're going to report any results, we like them to be an external data set and preferably prospective. So that way you get a really good judge on the generalization of this. So UCSF is one of the collaborators along with NYU and 2 European institutions. So this is some of the held out testing data set that we have for the 3 different predictions. This is IDH mutation, 1p19q, and then ATRX loss. And this is where the green line is the model that I've been proposing that seems to be allowing that these models are using the context as what is known of how these molecular subgroups co-occur and how specifically these mutations tend to co-occur in the context of the learned visual features from the stimulated Raman histology images. Okay. So that's, sorry guys. That's all I have. This is my group. I run the machine learning and neurosurgery lab at the University of Michigan. We have some fantastic collaborators at NYU, and it's been a fantastic road. This is still preliminary data. This is all unpublished results, and looking forward to see how this plays out over the next several months. So happy to take any questions.
Any questions from the audience? I obviously have a little conflict of interest here, having done a little bit of just a little bit of the mentorship on this project. What do you think is next to come in terms of our genetic classification based on the SRH images? How far do you think we can take this?
Yeah, I mean, I think it's hard. You kind of see how, I think, when we think about immunohistochemistry and well, I should say when we think about H&E histology and MRIs, we do know that humans can identify some differences between IDH mutated tumors and not. And it tends to kind of correlate with either contrast or enhancement or how aggressive the tumors look. So I think there is a precedent to know that there are visual features that are different between IDH mutant tumors. It gets really hard when you start thinking about these, CDK, TERT promoter, these much harder mutations where it's very much an open question as to how we can classify these things. I think the goal should be to try to do as much as we can, and I think incorporating what we all kind of taking a multimodal approach where we already know that there's certain way in which these things co-occur, combined with the visual learning that we can do with the network. So I'm not sure. It's an interesting open question from a computer vision standpoint.
Great. Looks like we have a couple of questions back there.
Hi, I'm Hannah Brown. I'm an analytical chemist at Purdue, and I do intraoperative work at Mayo. So I use DESI, desorption electrospray ionization mass spectrometry, intraoperatively to do IDH assessment. Our turnaround time is about 1 minute. I was wondering if you were I was curious if you could speak to the turnaround time of your method. I follow Raman as an alternative technology to DESI, but I was just curious if you could comment on that.
Yeah, so the image acquisition is the bottleneck. It depends on how big you want to image, really.
Yeah.
If we do our normal image acquisition, it's maybe a minute and a half or so. So it's on the same time scale, I would say. If you image more, it's going to take longer. The inference in the OR is very quick. We can run it on a GPU, so that's not a big issue. But I think these other sort of non-classic techniques, data acquisition techniques, I think they're great. I mean, I think with mass spec, you get very, very specific molecular data with that I think is a promising method.
Just a quick follow-up. Do you average across your, I'm assuming you're doing an image, right? So you're collecting, Raman information per pixel. Are you averaging, and then that's your representative IDH, or are you noticing heterogeneity in your IDH assessments throughout your image?
So the averaging actually happens over the model output. So the probabilities that we get out of the network is kind of where the averaging comes in. There's no averaging with the image acquisition component, but you're absolutely right that we do see regional differences. It's kind of amazing how infrequently the network is wildly confused. It's either really good and really right or really wrong. And I think that's really an artifact of your training set. If you're doing inference on an image that you haven't seen in the past, you're just going to bomb that one, most likely. Maybe you'll luck into getting it right. But in general, within tumor, I haven't seen there be wildly big swings in the prediction. So.
Thank you.
Yep.
Next question.
Yeah, this is Jay Zhu. I'm a neuro-oncologist from University of Texas at Houston. Again, I'm really excited seeing the presentations. One question is about taking this digital image of the pathology H&E stains. As we all know, the pathology itself is heterogeneous, even depending on how many pieces the surgeon gives to the pathologist. Mm-hmm. So how do you deal with that? You collect all the different H&E slices and then integrate together, or you take a sample of different areas of the H&E by the pathologist or with the training by the pathologist?
Yeah, so the surgeon, this is all happening in the OR, actually. It doesn't have to be. It's a bedside technique, so I think of it as a point-of-care technique. But in this case, it's great because the surgeon takes the tumor out, takes a specimen out, is able to image it. If it's a bad specimen, you can see that, right? We actually have models that predict that'll say this is a non-diagnostic specimen. If that happens, then just take another piece. So we have this ability to iterate in the OR that really can help the quality of the sampling, which is, I think, a bit what you're referring to. I think that's a big advantage of having this real-time feedback as to how good your tissue is. So I think, that's one particular part about it. The other part is you're absolutely right that it's a naive view to think that we should treat every region the same in terms of how diagnostic it is. That's actually a hard problem to solve because you can't really supervise it because you don't know a priori what the most diagnostic regions are going to be. I think that's a learnable function, and there are ways that people have tried to learn that, but it's an open question still as to how do we constructively build a system that's able to intelligently find regions that are diagnostic, but not have humans circle it because we don't know, right? That's part of the question. So,
Can I ask a second question? Do you have time?
Yeah.
this is a question that we all face in terms of brain tumor treating physicians. That's how to determine GBM progression versus pseudoprogression. I know Dr. Swanson mentioned that. I think the AI gives really a hope to address that, but I would love to hear some kind of answer, because we are puzzled by this every day, dealing with patients with MRI changes. We don't know the true progression, pseudoprogression. As Dr. Swanson said, waiting for 6 months, I mean, 6 months, patient's dead or, on the hospice pad.
Yeah.
So we really can't wait for that. So how do we address that?
It's still a ubiquitous problem. I think, anybody who spends enough time in tumor boards knows how this is, just continually an issue. It comes up time and time again. We've actually published on this, so differentiating, at least in our images, pseudoprogression versus true recurrence. And this was actually a really hard problem. This one really fought back because everything looks abnormal, right? It's all abnormal, even because of the treatment effect. So we kind of developed a way using our imaging modality, how do we differentiate this, and how do we use thresholding to determine whether this is true recurrence versus sort of quiescent tumor or treatment effect. So we were able to do that fairly well in our imaging set. But I do think that from a radiomics perspective, that's where I think it's really important, where developing these techniques is absolutely critical. I think it is going to take a multi-institutional data set to be able to solve that, because it's such a diverse problem. A lot of the pseudoprogression doesn't look, there's just a lot of ways in which people can pseudoprogress or recur. So that means you need more data to be able to represent those differences effectively. But I agree with you completely. It's a bad, hard problem.
Wonderful. Thank you to all of our